ISMB 2021 conference for giving us this opportunity to teach this workshop. Okay, so hopefully you can see my screen. We've got the workshop material for today here. And so your two instructors today are myself. I'm usually based in Peter MacCallum Cancer Centre in Melbourne, Australia. That's where I work, although I'm temporarily in Ireland at the moment. And Stefano Mangiola is the other instructor for today's workshop. And he is a postdoc at the Walter and Eliza Hall Institute in Melbourne, Australia. So I'll be taking you through the introduction, the workshop introduction, and also the bulk RNA sequencing part. And then for the second part, Stefano will be taking you through the single cell part of the workshop. And what we're going to be doing today is we're going to be showing you how you can perform analysis of both bulk RNA sequencing and single cell RNA sequencing following tidy data methods. And we'll explain a bit more about tidy data and what it is in a few minutes. And so during this workshop, we're going to show you how you can achieve this using these packages developed by Stefano that are available through the ANN, R, CRAN, and Bioconductor repositories. And they include these packages: tidy Seurat, tidy single cell experiment, tidy bulk, and tidy heat map. Okay, so for this workshop, there are some prerequisites, but we do have some introductory material for that to get everybody up to the same base. And this is the outline for today's workshop. So in total, it runs for 4 hours. So part one is going to be introductory background, and also bulk RNA sequencing. And as part of the exercises, there's opportunity for you to follow along running the code. We have an RStudio available in the cloud that you can use, and so you can run the code that we'll be showing you and see how that works. And we've also got some challenges that you can try out and a little multiple choice quiz if you want to have a go at the answers for it. And so the first part of the bulk RNA sequencing will be mainly focused on differential gene expression. And we'll also show you a little bit of cell composition analysis. And so that'll take about an hour and a half, and then we'll have a break for about 30 minutes. So you can get up, stretch, we'll get a drink, of water, or whatever. And then in the second part, we'll have the single cell RNA sequencing part. And that's Stefano will take you through this, which will focus on single cell analysis using tidy data packages. And we'll also have a little bit on pseudobulk analysis, and we'll explain what that is as well. And then at the end, the very last part of the workshop, we'll have time for questions and answers if you want to discuss things in more detail. But you are also welcome to ask questions as you go, so please type them into the chat. Stefano will be keeping an eye on that and can interrupt me if he thinks we want to stop and discuss it. And same when he's teaching, I'll be keeping an eye on the chat as well. So please do feel free to post any of your questions there. So yeah, so the format, as I said, is hands-on, running the code exercise with us, a few little challenges that you can try if you want, and then time for questions and answers. And so we'll be using the Zoom chat for interacting, and then we'll have this mentee quiz that we'll show you for if you want to have a go with the multiple choice answers. Okay, so this part describes the workshop goals and objectives. So the main aim being to show you how you can perform bulk RNA sequencing and single cell sequencing. So show you the main concepts for those, how you can perform them through tidy data methods. So following this tidy data paradigm. Okay. And this Google Doc here in this link, this has all the links you're going to need for today's workshop. And this has logins for the RStudio cloud. And I've just made that accessible just before we started the workshop. So you should be able to access that now. And it's also got the link and the mentee code for the little quiz we'll be doing, and also the link to the feedback survey. That would be great if you could fill out at the end and let us know what you think of the workshop. So I'm just going to open that up now in a new tab and show you, because it would be great if you could do this as well. Okay, so that's the material that we're working through that I already have open. And these are our studio logins. I won't get you to do that quite yet, but I will get you to go to this mentee quiz and to log in there using this code. So if you can, go here, pop in the code, and if I open that up, I'm just going to get that page over. Okay, so this is where we have a couple of questions for you, the audience, to get an idea of the background of the people here today. So if you can go to Vignette, yeah, some of you are already there, brilliant. So we just want to know, will you be running the code with us? Will you be trying out the challenges during the workshop? Or do you just want to sit and watch and listen, which is perfectly fine. Just gives us an idea to know how many people to wait for and to give a chance to have a go at the exercises. And then it helps us to know if you have much experience with transcriptomic analysis. And so that's either bulk RNA sequencing or single RNA sequencing, to know how to get a feel for how many people have transcriptomic experience, and also if you have experience with tidyverse. So are you familiar with the tidyverse? Do you code with the tidyverse? Also, that's brilliant. And we can see down here how many people have given us information here. So 18 of you have so far. That's great. Okay, so we can see most people are going to give it a go at coding along, trying out challenges. That's excellent, brilliant. Okay, some people have experience with transcriptomics, slightly more than, but not a massive amount. And then less people have experience with tidyverse. Okay, so you're here to learn about that. That is good to know. Okay, well, that's 19 of you. So that's more than 20 now. Okay, great. Okay, so it's staying kind of the same. That gives us, that's more than half. So that gives us an idea then. Okay, so a bit more explanation of tidyverse, and some, but not everybody is familiar with transcriptomics. That's okay. We'll give you a bit of a background on that then. Okay, cool. So that was this questions here. And so this was asking you to rate these questions. Okay, so now we have a little bit of background before we get into trying any code. So then what is transcriptomics? So that's all the RNA transcripts in the cell. So in this picture here of the cell, the transcriptome, you can think of it as a bridge or a connector between the genome that has your DNA and the proteome, where your proteins are made. So these are the nucleotide messages that get sent out into the cell, and they can get turned into the proteins. Or we also do have some transcripts that don't code for proteins, but that they have their own functions, so regulatory functions within a cell. And we have many thousands of genes, many thousands of transcripts that can be made by an individual cell. And different cell types will make different transcripts. Okay, and why do we use transcriptomics? Like, why do we study transcriptomics often more commonly than genomics or proteomics? Because the genome tends to be pretty stable. The transcriptome is usually what's reacting if you, say, apply a drug. It's the expression levels of the different genes that will be seen in the transcriptome. And we could measure the proteome, but that's a bit harder. And as I mentioned, not all transcripts code for proteins, so you may also be interested in non-coding transcripts as well. So studying transcriptomics, that allows you to measure changes in expression levels of thousands of genes of both coding and non-coding. And this is an example of a type of RNA sequencing experiment that you might be using to generate transcriptomic data. So you may have some samples here that are being treated with a particular drug, and you may want to know how this drug works, and you may be measuring the transcripts from these cells and comparing them then to untreated samples. So you want to see which genes, which transcripts are changing in their expression level, in the amount that's being made. And that can give you an idea then of what the drug is doing. So that's one example of a possible experiment. And how does it work? So with transcriptomics, what happens in the lab is that you take your cells, you extract your mRNA, we call it the messenger RNA, your transcripts from that. So this is representing 3 different transcripts. And we shatter them into small pieces and then put them in a machine called a sequencer, and we generate these short sequence, what we call reads. So these will normally be about 50 to say 150 nucleotides in length. So we've got all these little pieces. And then we can do different things with that. This would be our raw data. And we can then do a differential expression analysis. So that would be the most common type of analysis in transcriptomics, where you have 2 groups, like I said, treated and untreated, and you want to know what are the genes, what are the transcripts that are changing. And another type of analysis you can do is this cell type composition analysis, which we'll show you a bit in this workshop. And that's where you can try and determine what cell types are present in your RNA sequencing sample. So you could measure, for example, the amount of different immune cells. Like, do you have more or less B cells, T cells, things like cells like that in your 2 different groups. Okay, and then there's other types of analysis you can do, but we won't be covering them in this workshop. Okay. And so this then is an example of a type of bulk RNA sequencing differential expression pipeline or workflow. So what happens is you take the raw sequence reads that come off that sequencer, you then map them to a reference genome. So for example, to the human reference genome, to see where do these little pieces come from, which genes were producing them. Then you count how many reads you have for each gene. And then you go on and explore the data, check it for quality, producing different plots to see the good quality data. And you then perform the differential expression. So comparing your, for example, treated versus untreated groups to see which genes, which transcripts are changing. And then you can go on further and do things such as pathway analysis, which is seeing which groups of genes, which pathways are changing in your groups of interest. And so in this workshop, we'll be showing you how you can do this with for bulk RNA sequencing using tidybulk. And then in part 2, we will be going on to look at single cell RNA sequencing data. So Stefano will take you through this. So he'll come back to this then and to the single cell workflow. But for now, we will switch to say, okay, what is tidy data and the tidyverse? Because that's what this workshop is focusing on, how you can analyze this type of transcriptomics data in a tidy way. So this tidy data paradigm was something that was developed or started by Hadley Wickham some years ago. And what it does is it provides a standard way to organize, to format data sets. And so this here is trying to explain it. So with tidy data, every variable has its own column and it's just present in one column. And every observation is in one row and every cell is a single measurement. So you're not bunching things together. You don't have data in like the same variable spread across multiple columns. And this is all about trying to have data in a nice format that then you can more efficiently analyze without having to write huge amounts of codes to format the data and to wrangle it and get it into the way you need it before you can even start doing the analysis of the data. So it can make your coding and your analysis a lot more efficient. And the tidyverse then is a collection of packages that were established by Hadley Wickham. And you can use these to tidy your data and manipulate it and get it into this tidy data format. And you can also visualize it then quite nicely. And so in this workshop, we're going to be using many of the functions from the tidyverse package collections. So these are functions that you're going to see, so such as filter, we filter rows, select for columns, mutate for adding columns, pivot longer for reshaping, and then ggplot is a hugely popular package for visualizing data. And so these packages and functions are all pre-existing. And then what tidytranscriptomics packages that you'll see are doing is building on top of these existing nice packages, structures, ways of analyzing your data. And so this is just an image then showing there's these different packages that belong in the existing tidyverse collection of packages. And here we'll be using a few of these manipulating functions and also these visualizing ggplot functions as well. So that's just a little bit of background for you. And what we're going to do now is, for those of you who want to try out running the code, we will get you to get set up using one of these servers. So this is the easiest way to run this material during the workshop. We've kindly been given this RStudio servers in a cloud for you to use. So there's logins for the servers in the spreadsheet that I'll show you in one moment. So we'll get you to take one of those logins, write your name into the spreadsheet, decide which one you're using, and then open up this file here that's in this folder there. And then after the workshop, so this will only be available for today's workshop. It'll have to get shut down then. And after the workshop, you can, if you want to use this material, you can install it on your own computer and you can find the instructions here in this link here. So if I just show you here, this will explain how to set it up on your own computer. So afterwards. And this material, this website that I'm showing you now, this will remain available after the workshop. So you don't need to worry about that being gone after today or anything. And if you just want to sit and watch, that's fine. And that link there is just pointing you to this workshop page that we've got here. Okay, so now we'll take a few moments to get everybody set up with the RStudio login. So if you go back up to the Google Doc here, so this Google Doc, which I've got open here, this is the RStudio logins here. Everybody's got one. So if you click on that, I'm going to open it up here. So I'll show you that. Okay, so we've got 3 different servers, RStudio 2, 3, 4, and we've got 30 logins available on each. So you can spread yourselves over them. You can take whichever one nobody's using. So that's RStudio 2, RStudio 3, we've got RStudio 4. So feel free to take whichever one you like. And so this will be the username to log in. This is the password. So, and if you can write your name beside which one you're using, that would be great. So no one else tries to use that, because only one person can be logged into each individual username at a time. And what you need to do then is I will open up my one. Oop. Let's grab that link, copy it. Okay, so for me, I'm going to get set up here with this is my password. And if anybody can't find this spreadsheet, let us know and we'll put it in the chat.
Yeah, I will share it now in the chat.
Oh, brilliant. Thanks, Stefano. We'll stay signed in. Okay, so when you sign in, this is what you should see. Well, I have it already open because I was checking it worked. So you should see an RStudio interface like that. And excuse me, it should be in the files pane, this ISMB folder in here. There should be a vignettes folder, and then this tidytranscriptomics RMD is the one to click on to open it up there. And what would be great is, are we able to? I was just seeing if we can, in Zoom, have that option to have the green tick if you have it ready, if you're at this stage, but I don't see that option in my view.
I think roughly half of the user have put their name. So I guess
Okay.
if
Yeah.
help, let us know, and we will wait until everybody that wants to follow along. I suggest everybody would do it.
Oh, I don't know whether, can you see the message that's popped up? I think that's because somebody else is, no, it might be myself because I had it open in another window. So that's what'll happen, the disconnected if someone else tries to log in, but that's okay. Just click off the other tab that I had open and refresh.
Yeah, please
okay.
make sure you don't log in to
Just someone else.
any account already taken, because if you do it for hours, it will crash for everybody to see, but you will crash whatever account is already used.
Okay, great. So we'll give you a minute to get started. Stefano has put that in. Thank you, Stefano, in the chat. Oh, I'm getting another disconnect. So remember, yeah, do not use one if there's a name here, because you'll kick that person out. But take any of the other ones down here that you like.
Yeah, if you have to choose, take server 3 and 4 because they are the most free.
Yeah, exactly.
So
it's good to spread out for memory reasons and things. Like, it should be all fine from the testing we've done, but good to spread out. Oh, training, someone's saying training 308 is not working. Okay, yeah, please take another one if it's not working. RStudio 3, if you're having an issue with that one, try RStudio 4 and I'll have a quick look. Like, we should have plenty. There's like 90 logins here, and so there's only 35 people here, so. Miss hyperlinked. Let's see. Oh, is the hyperlink not working? Let's see. It works for me. Is RStudio 3 hyperlink. Is anybody having issues with that one? Try RStudio 4 anyway, yeah, if you're having any issues with 3. Or let us know. Can you show where's the RMD file to open? Yep, absolutely. So when you log in, you should hopefully see this. Do you see this? You might even be one level, yeah, in this folder that says ISMB 2021. And then in the vignettes folder, here, this tidytranscriptomics.rmd file is what you want to open. And if you click on that, that should pop it open in the file editor pane here in the top left. Is that okay, Dina? Because if anyone else is having trouble with that, please let us know. Or if anyone's having trouble with the logins, please error unauthorized. Which
Is there anybody with working server 3 or 4? Because it seems there are a few problems. So if just one person confirms that server 3 or 4 is working good.
Oh, I see what that, if I hover over the link, yes, it's saying 2. Hold on, they're right. So I think the text that's written is showing 3, but it's actually bringing them into 3 and 4. Let me fix that.
Yeah, so copy, yep. I think
okay.
You can copy the text above anyway. The link.
Edit link, okay.
Text.
RStudio 3, yes. The text is saying 3, the link is saying 2. Thank you for flagging that. Okay, and I'm going to copy that all the way down. Okay, so RStudio 3 is now fixed. Apologies for that, and I'll fix RStudio 4.
Well, all packages should be installed in all servers.
Yeah.
Somebody's saying that tidyverse is not present in server 2. Okay.
Tidyverse is not. Well, we've installed the individual tidyverse packages, if that's what you mean. You can't do library tidyverse.
Yeah, so in the file, RMD file, there are all library needed. I think, yeah, as Maria said, we probably don't have tidyverse, but the single packages.
Okay, so okay, people are saying great, server 4 and 3, or well, 4 anyway. Yeah, okay, and yeah, that answers your library tidyverse. Yeah, no, we installed them individually. Okay, hopefully anyone else having issues now, let us know. So the links should be fixed. I'm going to reconnect. Oops.
Great.
Works swimmingly, great. Okay, 12:33. How are we going? Would people, is there people who'd like another minute or two? If you can, let us know in the chat. Or server 4 is working, just a few libraries aren't loading. Let's see. Well, let's check. I'm going to do the library load in a minute anyway, so if people, I might do that now, and then people can let us know if you're having issues or not, and we can see what's happening. Because worst case, you might need to jump from server 4 to server 3 if there's any issue. We did test across these, so it should be working. Okay, so okay, I might continue on then, and let's see if everybody can load the libraries. Okay, yeah, and shout out in the chat if you're having any issues. And if you are having severe issues, and Stefano and I can help you on the break as well. So don't be worrying. Okay. So what I'm going to do is, hopefully most of you are seeing this screen here now with the tidytranscriptomics open. I'm going to click on this little icon at the top, which gives this table of contents along the right-hand side of, this is what's called our Markdown document. And I'm going to jump down to where we were. This is the Markdown that becomes the website I was going through. So we were down here getting started. So I'm going to click on this, what is tidytranscriptomics? Okay. And explain this. Then we'll load the libraries and see how everyone's going. Okay, and I'm going to just adjust my screen a bit to make that a bit bigger for you. Hopefully, if you're having trouble seeing, let us know in the chat as well. Okay, so what is tidytranscriptomics? So I already mentioned that it is a collection of packages designed for analysis of bulk RNA sequencing and single RNA sequencing. These are the main packages linked here. And these enable you to perform analysis of the RNA sequencing data in a tidy way. And this image here is a roadmap of the development of these tidy packages. So Stefano, who's the other instructor today, is the developer of these packages. And this image is showing what's been developed so far and the future plans. And there are packages for the bulk RNA sequencing that I will focus on, and then Stefano will focus on the single cell sequencing. So for the bulk RNA sequencing, one of the packages that I'll be showing you in a few minutes is this tidy summarized experiment package. And what that does is it enables a tidy representation of data that's in summarized experiment format. Summarized experiment format comes from the Bioconductor, if you've heard of that, the Bioconductor community, Bioconductor repository of packages for RNA sequencing analysis. So that format is not a tidy format. So this package here enables you to work with the data using familiar tidyverse style functions. So I'll show you that in a moment. And then the package for bulk RNA sequencing workflow package is called TidyBulk, and we'll also be showing you that today. So that's how you can perform actual RNA-seq analysis steps. Okay, and then Stefano will explain these packages here in the single cell part 2. Okay, so with RNA sequencing, I showed you that workflow before, of the steps of an analysis, from reads to mapping to a genome to getting to counts, and then performing analysis. So for today, we're going to be starting from the counts, when you have counts of reads for each gene, and we're going to be starting from there and performing the analysis. So the first thing we're going to do is load the packages that we need. So what I'm going to be doing is clicking on this little green arrow, which runs the code in what we call a chunk. And you can do that too as an exercise to follow along. So I'll run a chunk and then I'll explain what's happening and what the chunk is doing. So in this case, we're loading all the packages that we need. So our data set is the tidyverse packages, and then these tidyverse friendly packages. So So here we are getting the messages that the packages are all loading. And so lots of red messages, but that's all fine. So it would be great if you could try that. And if anybody who's on RStudio 2, 3, and 4, if one person from each could let us know if that's working okay. Do you see this? So we can see are all the libraries. Okay, 2 is good, brilliant, thanks. 4 is good, excellent. What about 3? 3 is good, excellent. Okay, but if anyone is having issues, like I said, feel free, post in the chat or we'll give you help on the break. Okay, excellent. Okay, so next chunk down then is the colors. We're just setting the colors we're going to use and also a theme for our plots, for our ggplot. So I'm going to run that just to make our plots look a bit nicer. Okay, but then the data. Okay, so the bulk RNA sequencing data we're going to use today is from this airway package, and it's from this paper from Himes, and it's 8 samples from human airway smooth muscle cells. These samples are from 4 cell lines, so each cell line has a treated and an untreated sample. So the treated sample has been treated with this dexamethasone, and the untreated sample is the negative control. Okay, and the samples underwent RNA sequencing, and we have here the gene counts. So this is how we can load in the data from this existing package, and we can have a look at it. And so this format is that summarized experiment format that I mentioned. So it's not in tidy data format. This is how it's represented. So it can be a little bit tricky to see what's actually inside if you're not familiar with this format. And if you're interested in the summarized experiment, you can find more information on it in this link here, Bioconductor. And what we're going to see now is this tidy summarized experiment package from Stefano, what that helps with. So here I'm going to load that package to show what that data, airway data now looks like. Okay, so remember, so airway data, we printed it here, it looked like that. Okay, now we've loaded the tidy summarized experiment, and now it looks like this. So this is tidy format. Now we've got a column for every variable and a row for every transcript, so for every gene. So this makes it easier to see what's in this data set. And this is telling you that it's a summarized experiment tibble abstraction. So it's still summarized experiment format, but it's enabling you to view it in a table form. So tibble is the tidyverse name for table. And here we can see how many rows we have. So we've got 500, over 500,000 rows and 13 columns. And we can see how many genes, how many transcripts we have. So we've got 64,000 transcripts, and we've got 8 samples in this data set. Okay, and our first column, we've got our gene IDs. So they're ensemble gene IDs. Our second columns are sample IDs, and then we've got the counts. So this is how many reads, how many counts we have for each gene. So this is the expression level that we're going to be working with. And we've got a column that tells us which cell line. So we've got 4 cell lines. And then we've got a column called dex, which tells us whether the sample is treated or untreated. So they're the columns we'll be working with. And with this tidy summarized experiment object, we can still work with it using summarized experiment commands. So if that's what you're familiar with as well, you can use those on this data set, commands such as assays. Here we're going to be focusing on the tidyverse commands. Okay, so now we're going to show how you can use some of these commands that I mentioned before. So filter, select, group by, that come from the established tidyverse packages. So if I'm going to run this, so here I'm taking that airway data set and I'm using filter to select the treated samples, the treated rows. So I'm saying, give me the dex column where it says treat. So now we've gone to 4 samples using the filter function. And if we want to choose particular columns, because filter is where we choose rows, we can use select. So here what I'm saying is I would like to have a look at these three columns, the sample, cell, and dex. And I can do that using the tidyverse select function. So here we go. There's our columns. And we can also use tidyverse group by and summarize if we want to calculate the total counts for each sample. So here I'm saying group by. So for each sample, get me sum up all the counts so I know what are the total counts for every sample. Okay, so I can do that. And you can see they've all got around 20 million. And if we want to create a column, we can use tidyverse mutate. So I'm just going to run this. So what this is doing here is it's saying for the sample column, so see the sample column, they've all got this prefix, but the unique identifier for the sample is actually the last 3 digits. So I'm saying, and this is using, again, standard tidyverse method. So I'm saying remove this prefix from the sample column and use mutate to create a new sample name column. And then I'm selecting the sample and the sample name column to have a look at those. Oh, and I forgot to mention that here we're using the pipe a lot, and this comes from tidyverse. So this is an existing tidyverse way of chaining together commands. So this can enable you to, like here, combine different commands without having to keep saving as intermediary objects, and can make the code easier to read. So you can see I'm doing this step and then I'm doing this step. So this pipe is taking the output from the left and piping it into the command on the right. So here what I've done then, yeah, I've created a new column with mutate that is just without the prefix. Okay. Okay, so the idea there is to show you how this package, tidy summarized experiment, enables you to work with data if you've got it in summarized experiment format, to work with it using existing tidyverse commands, which is nice. And now what I'm going to go on now and do is set up this data to actually perform the RNA sequencing analysis on it. So what I'm going to do now is to take that airway data set that we had, and I'm going to do what we just did there, which is create a sample name column where we strip off that prefix. I'm going to then add, this is to get the gene symbols for the genes. So at the moment, we've only got the ensemble gene identifiers, but if we want to know the name, like which is the P53 gene or whatever, we like the human readable names. So I'm doing that here, and I'm saving it as an object called counts. So that's having a look at it here now. So I should see that now I've got 2 more columns. So went from 13 to 15. Now I've got a column for sample name, and I've got one for symbol as well. Okay, so that's formatting our data. Now what we're going to do is the steps for an RNA sequencing analysis, which is here we're going to get rid of genes with low expression. They don't provide much information for our analysis. So we are going to get rid of them. I'll run here. Now we're using the tidy bulk package because we're actually performing a bulk RNA sequencing workflow. So I'm using a keep abundant function from that package. And for that, I just need to specify the column with my groups of interest. So here it's the dex column that says whether samples are treated or untreated, and that will filter out genes, transcripts that have very low expression across most samples. Okay, and here now by running that, we have gone from, we were at 64,000 genes. We have gone down to now about 15,926 genes. So we've gotten rid of, some of those will be genes that have 0 counts across all samples. Some will have just a couple. So we've gotten rid of them. And the next step we're going to do is to scale the counts. So this is also a step we perform in RNA sequencing analysis, where we need to remove the differences that are not interesting between our samples. So they can be technical differences due to sequencing different amounts for different samples, like maybe you did double the number of, generated double the number of reads for some samples versus the other, but that's not part of the biological difference in expression. So you want to get rid of differences in what we call sequencing depth and also sequencing composition. And you can read more about that at that link here. But here for today, to know that Tidybulk has this scale abundance package, which will scale the counts, will normalize the counts, and it has a number of different normalized methods available, but we're going to use the default one, which is a commonly used one called, from a package called EdgeR, trim mean of M values. We're using that scaling method. And the output from running that function gives us this column called counts scaled. And so now we have our counts for each of our transcripts in each of our samples. We've got scaled values. Okay, so the nice thing about, Tidybulk enables us to use tidy methods, so tidyverse ways of analyzing data. So this here, which I'll run, is showing how we can quite easily use existing tidyverse functions to shape. Like pivot longer is a great function, very powerful for reshaping your data into a format for then plotting, for example. You'll see us using that more throughout the workshop. And so we can quite simply take the data we've got, pipe it into this existing function to reshape, and then generate some ggplot, so some existing tidyverse plotting functions to, with a few lines of code, generate. Here we're generating plots of the distributions of the counts before and after scaling. So this is a line for a density plot for each of our 8 samples. And we can see that after the scaling, the distributions are more similar. We don't have any really weird looking samples. So that's quite nice. And so for comparison then, so this is how we can join the steps we've just done above, and to show you that it's actually just a few lines, relatively few lines of code to take the data, to filter out the lowly expressed genes, scale the counts, and to generate those density plots to get a view of the data. It can be done in a few lines of code compared to the more traditional older ways of analyzing the RNA sequencing data. For example, here using a package such as EdgeR. So this is to show that to do something similar, to take the data and generate density plots before and after scaling, you'd have to write a lot more code, create more variables, more room for typing errors in the code, and also some of the functions may be harder to understand. Because another feature or another aim of tidytranscriptomics is to provide functions that have more easily understandable names. So names such as filter abundant, scale abundance. So it's easier to understand what they're doing. Okay, so another type of plot we generate with RNA sequencing data is a PCA, principal component plot, or a MDS, a multi-dimensional scaling plot, to check if the data looks as we expect. And so we can generate these plots using Tidybulk package, so generate them in a tidy way. So I'm going to run this here to show you. So tidy, and the reason we generate these plots is we want to see, do our samples group as we expect? Are we seeing, one group for treated, one group for untreated? and are we seeing any outliers in the plot? So this is a really important way to check, essential way to check your RNA sequencing data. And so here, tidybulk has a function called reduce dimensions. And here we're using the PCA method. You can also choose the MDS method. And this we'll go on and plot this data in a moment, but just to show you that it will give you the 2 columns by default, and it will give you the fraction of variance that is attributed to each of these principal components. So here we can see the fraction of variance for principal component 1 is 0.35, and the fraction of variance for principal component 2 is 0.31. And we have our first little challenge question, which is what fraction of variance is explained by PC3? So this is something that we have a multiple choice option in the Mentee quiz. So it would be great if you could have a little go at that. I'll give you a minute and have a go at figuring out what is the fraction of variance for PC3. And if I move on in Mentee, okay. Okay, oh, shows all of you in it, great. So, okay, it should pop up now again with Mentee using the same Mentee that we're already using. So we've got 4 choices for you. Here is PC3, is the fraction of variance explained by PC3 0.31, 0.27, 0.18, or 0.16. So if you can vote on which of these you think it is, and we'll stop it in a minute or so once a few people have had a chance to have a go at it.
Put on the chart, but that's pretty easy. Go on www.mentee.com and you will be asked a code, and the code is visible on the screen, but also in the chat. And this is the platform we will use throughout the workshop. So stay logged in, and you know, your session will continue. There is a challenge, and at the end we will see there will be a winner. Maria will think about some present to send.
Right. Okay. And if you don't try the code, you can also just guess, because it is multiple choice, and just, could you share the Google Doc link? Yeah, will indeed. Or
if you click share, it's safer. I'll do it.
Oh yeah, I was going to say, because I can keep this open then, so we can keep an eye. So we've got 9 people who've had a vote so far. Cool. And if you have any questions as well at this point, feel free to ask them in the chat. Or if you want us to go back over anything, or.
A little tip is you can read the documentation of the function we were talking about. Put in the question mark before the function name, and there will be an argument that will say probably what you want to know. It's pretty straightforward.
Someone's saying, I am the only one where it just says what fraction of variance is explained by PC3 5.
Just says there's no option to select.
I only see 5. So it's because 14 people have been able to do it. So why is it not working for?
Only see 5. I don't see any answer and option to select. Okay.
But so this is in the mentee, is it? Yeah, for that you're only seeing, yeah, mentee.
Okay, that's unfortunate.
And for the people who are having trouble, what about, can you do it on your phone, or are you already
What could be 5? Do we have any question with 5 as answer?
Unless you need to press enter or something to get onto the next page, because there's 5 questions. So unless it's telling you there's 5 is the time to see the alternatives.
No working on my phone also. Wow, that's unfortunate. It's fine. Wow, okay.
Same. Okay, that is
Let me do
very
it just
weird.
for the sake.
Yeah, I'm just going to try it in an incognito window as well to see what happens. Oh, I see what you mean. Yeah, I'm getting that as well. What fraction
But
of
fraction of
variance?
variance 5. Wow, okay, that's so strange. Yeah,
5.
We tested yesterday for some reason.
And it's working for some people, but not others. That's very bizarre. I'm just going to check.
It's fine. So
yeah.
If it keeps to be problematic, obviously we will go to the more simple version, which is stream the answers to the chat, and we will do a question by question challenge, and we will be able to see kind of the statistics as well. But you trying to do the exercise is the main thing, and then to put the answer in the charts, and we will reveal the right one. All right, people started to put their answer. Yeah, I think we can reveal the answer.
I think we can reveal as well. Okay. Okay. Oh, interesting. I don't know if anyone else saw that, but if you were having that, what is the question 5? It looks like because it had already started, it wasn't letting you log in. And now I'm getting an option for putting in my name. So the answer was indeed 0.16. So those of you who popped it in the chat as well, correct. Okay.
Go cake.
Go cake, yeah, fastest.
Yeah, so stay logged in, and probably will work.
And anybody who's having what is the answer 5, try logging in now. I'm getting the option to put your name in, so I'm gonna see if I can join. And then hopefully it will let you vote on the next question. Yeah, I can put a name in. Okay, let's see. Hopefully if it lets you do the next question, or else drop it in the chat. Okay, let's go on. Well done. Okay, oops, I'm on the RStudio server. Yep. Yep, that is it. So I might just pop in the solution here. So it was count, scales, reduce dimensions. Oh, my typo. Method equal P CA, and then it was dot dims equal 3. So that should, oops, get you the answer. And how to see that is you could use the help. So the question mark, reduce dimensions. And if you had had a look at the help, you should see that there is this dot dim. So you can see more than the top 2 dimensions if you would like. So great, most of you got that. Brilliant. Okay, and so moving on then, what we should see is that what we did up here by running reduced dimensions and saving it as the counts scale PCA, what has happened is the principal component information has been joined on as columns to the counts objects. So we should see here we've got the list of the 11 more variables columns. So we now have one for PC1 and PC2, and I'll show you what it looks like in a second. So with this object here, we've still got our one row for every transcript in every sample. But if we just want the principal component information for the samples, for the 8 samples we've got, we can extract that using this tidybook pivot sample information. So if we run that, okay, now we should see that we've got 8 rows and one for each sample. And if we click along, we should see here is our principal component 1 and 2 values. And this is what we can now then plot. So if this will generate a PCA plot for us. And here we're showing again that you can, because this is all in tidy format, you can pipe it all nicely into ggplot and generate a nice customizable ggplot from it. So here what we've done is create a scatter plot with ggplot. We're coloring by the treatment, so treated versus untreated, and we're using the shape. So we've got different shapes for the points for the different cell lines, for our 4 cell lines. And then we're using a tidyverse friendly package called ggrepel to be able to repel the names of the samples away from the points so we can see them. So this is what your plot should look like. And we're seeing this data set looks nice. The greatest amount of variation on PC1 is separating our samples into our treated and our untreated groups, which is what we hope to see here. And we can also see, though, that one of the cell lines, the cell line that's a square, is a bit more different to the other cell lines, the other 3 cell lines. So that's this cell line here. Okay, and our alternative way to view the relationship between samples is hierarchical clustering. And we can do that nicely with these tidy packages as well. So I'll run this again and explain. So what we can do is take our counts. We can use this tidy bulk function called keep variable to extract the 500 most variable genes. And we can then create a heat map using the tidy heat map package and color by our different treatment and cell lines. And I see a question in the chat saying, what are the numbers close to the points? These numbers, so they're the IDs of the samples. So that was when we were creating that sample name column. This is sample ID 520. Like you might have more informative names like, I don't know what the actual cell line or whatever your ID for knowing what that sample is. So that is how we're labeling here with the sample name. Okay, why we see so few points? Okay, that's because we've only got 8 samples. So if we were using a bigger data set, you might have a data set with hundreds of samples, then you would see hundreds of points. So one per sample. This is for the sake of running a workshop, time allowed, resources allowed. We've got a small data set here, but the principles would be similar for larger data sets. Okay, and so if we generate a heat map of our most variable genes in the samples, here we see, these are our 8 samples again. We've got one column for each here. And these are the 500 most variable genes. But here what we want to see is how are they clustering? How are they grouping? And we have added this annotation colors for which cell line they're from and which treatment. So here we can see, like with the PCA plot, our treated and our untreated are grouping together, but there's one cell line. So this yellow cell line, that one there, is a bit more different than the others. So like what we saw with the PC plot as well, PCA plot, that it was further, that cell line was further away than the others. So there's a bit more variation there. Okay, and here we're showing again that if you weren't using these tidy packages, this would be an example of writing this with traditional R ways. So for example, using edge R. So there is more variables that you'll need to create, some functions that may be harder to understand the names of. And okay, so now we've had a look at our data and it looks quite nice. We can see there's a bit of variation due to that cell line, but we can go ahead and perform our differential testing. And a really nice thing about TidyBulk is that it's integrating several different RNA sequencing testing algorithms. So some of you may have heard of them. So there's the EdgeR mentioned before. There's LimaVoom is available, and DeepSeek2. And these are all very good algorithms for performing the testing, for seeing which genes are different in your groups. So with TidyBulk, the function for testing is called test differential abundance, and you can run one or more of these methods depending on if you want to compare them or not. So I'm going to run this and explain it. So here we're taking our counts object. We are performing the testing here. For all these testing algorithms, we provide a formula. So we say which column contains the groups we want to testing. Here it's DEX. We also know there's some variation due to the cell line. So we're adding that as an additional factor. And then we're specifying which method we want to use. So here we're testing EdgeR quasi-likelihood. We're also going to test the other method, LimaVoom and DeepSeek2, and we can quite easily compare them or run them all with TidyBulk. Okay, and so that has run there. And now we should see that we've got these columns here added for EdgeR and DeepSeek and Voom outputs. So these are the columns that give us our log fold change. So the amount of expression change for that gene in our treated versus untreated. And we also get false discovery rates and p-values. And here are it's our log fold changes for treated versus untreated. And we can take advantage of I'll run this tidyverse friendly packages such as ggalley to quite easily create pairwise visualizations of the methods so we can see which ones are more similar to each other. So here I'm extracting the transcript information from the results. So one row per gene. And I'm selecting all the p-value columns for the 4 different methods. And then creating the pairwise plots. And so what we can see here is that these 2 methods, EdgeR, QL, the 2 EdgeR methods are the most similar. And we can see here that the ones that are a bit more different from each other are Voom and DEC2. So comparing the p-values for the different methods. Okay, and here we've got a little challenge for you again. So with our mentee, if you want to have a go at this, which of these 4 methods detects the largest number of differentially expressed genes? So using the p-value adjusted for multiple testing, p-value less than 0.05, which depending on the program is FDR adjusted p-value or p-adjust. Okay, I'm just going to get that. So this is another mentee one. So hopefully you can access that this time. Okay, I'm going to start. We'll give you a minute or two, a couple of minutes to do that. And I can see, is there questions in the chat? Is the heat map function used from tidyverse? No, it's from tidy heat map package, another package developed by Stefano that's very popular. And it's on CRAN, yep. How do you make sure tidytranscriptomics stays up to date with new versions of, for example, EdgeR? So tidy bulk, so the package that's using EdgeR, DEC, etc., is going to pull in the most, is this right, Stefano, the newest version available. It's going to use whatever you have installed for EdgeR. It's going to check if you have EdgeR, Limma, DEC2 installed. If not, it will pull in the newest version. Is that correct?
Yeah, they are just dependencies. It's just a wrapper of these packages that build up a uniform, a consistent framework in a sense. If this is the question, it also happened that Limma changed how MDS kind of output the results. So we obviously try to keep, when that happened, there are pretty established packages, I have to say, but when that happened, of course, we update the backend.
Yeah, thanks. Yeah, I was also wondering, for example, maybe for bulk RNA-seq it's quite established, but for single cell packages, things are changing a little bit more, I guess. So I was just wondering, so sometimes if something really changes, so for example, Surat brings out a new way to integrate ATAC-seq with whatever, in that case,
Yeah,
you'd have to manually update your packages, I would say.
Yeah, I mean, obviously, tidy bulk is just for bulk. I will show you later single cell, and I will show you how we, with our packages, we do just data representation. We didn't implement a workflow yet, and we will see in the future if we implement it. So that problem in this case will not apply, but we can discuss further later on. But yeah, so good.
Great, thanks.
So since we are waiting, a user wrote to me, and I would like to say more, but I prefer to speak rather than. So the question was why in the heat map often there are so many genes or feature plotted, and often you cannot even read the gene names, and you have such a big amount of information that what sense does it make? And so for once, a scope of the heat map could be to see how the sample cluster and to have a very overall representation of your genes distribution. So you, first of all, you might not even care which gene is which. So you might just use it for the dendrogram representation, hierarchical clustering. A suggestion is to not plot all feature, but to be unbiased. To start with, you plot just a variable feature, and there is a function in tidybulk called keep variable. So you would get an unbiased clustering of the samples, but then to prove that after you have done the differential expression analysis, to prove that at least there are some features that cluster your sample, you can even filter based on the significant genes and redo the heat map. It will tell you a different thing, but you will have much less genes. Apply some filtering. That's the answer.
I might just go till 11. We're like 11 minutes left, so 50 more seconds, and then we can move on.
Who is able to use Mentee, please do. Is a great way to interact. Plus, soon here it will be midnight, so if you don't interact, we'll fall asleep before it's my time. So.
Or if you're having trouble with Mentee, feel free to use the chat to put in which answer you think it is. Okay, I might just, in the interests of time, okay, I'm going to give 10 more seconds. 8, 7, 6,
yeah, this
5,
is also a hard
4.
question, actually.
This is a harder one.
Yeah, some people might need more time.
Okay, but you can go back, in the interest of time, I'll move along, yeah, to make sure we get our break in. Okay, so well done to those of you who got it. And the answer was, hopefully you can see, was the edge-or likelihood ratio. And Frank was super fast there, so well done, Frank. Okay, and if I go back to, well done everyone for giving it a go, or feel free to give it a go afterwards. And in the interest of time, again, I'm going to just paste in the solution. So you don't have to watch me with my typos. So this is one way to do it. You might have other ways. This is a very tidy way, a tidyverse way of doing it. So you can take the data, get the transcript information. You can reshape the method output columns using the code here. And you can then filter for the statistics, so the adjusted p-value rows, and then look for the values less than 0.05, and then count the rows for each method, and then sort them to see which one has the most. So that's one way to do it. So if you do that, so you can see edge-or likelihood ratio has the most differentially expressed genes with nearly 5,500.
So this is a decently elegant but very complex way to do it. I agree. Mm-hmm. Another simple way is to do for filtering. So not go in one, but filter for, sorry, not filter, but filter 4 times for each of the statistics, full discovery rate and so on. And every time ask, filter for full discovery rate less than, and see how many you got. And then you get also your results much simpler. Yeah,
that's probably how I would do it. That was Stefano's solution. But whichever way it suits you, feel free to use. And okay, so you can also, that was showing you how you can run multiple methods if you want. You can also just run a single method. So for the test differential abundance, so by default, it will use the edge-or quasi-likelihood. You can choose which is your favorite. And so again, just to say, this would be how you would do that testing using traditional R. And as you can see, again, more variables you'd need to create and harder to understand, arguably harder to understand function names. Okay, so now we're just going to show a couple of plots you can make after you've done your testing. So a very common one people like to make is what's called a volcano plot. And this code here shows how you can make a minimal type volcano plot using here. So this is plotting each point is a gene. And this is showing the genes that are upregulated in our treated samples. To the left of the 0 are the genes that are downregulated in our treated samples compared to the untreated. And we've got the most significant genes at the top. And a nice thing about TidyBulk is it takes advantage of the ability to have custom ggplot scales. So it provides this log 10 reverse. So instead of seeing the negative log 10 value on the y, which can be hard to interpret, you can see the actual p-value by using that. So that's a nice handy feature. And if you want to create more, say, fancier plots, customize them further, for example, you might want to plot the top most significant genes. You could extract, this is showing how you could extract the top most significant genes by p-value, top 6. So we're using tidyverse functions here. So pulling out the names, symbols for our top 6 genes. And then we can take advantage of ggplot's ability and tidyverse ability to customize our plot even further. So we can label color our highly upregulated and downregulated genes. That's what we're doing there. And we can apply some more just visual customizations to the plot, highlighting our top 6 genes. So it can be a nice way to take advantage of the ggplot tidyverse ability.
Just to point out that we are showing here some example of how to do custom plots in a quite simple way. Of course, there are plenty of methods out there, and I suggest you to explore them to do this plot with one line of code, and there might be some argument for customization, but the power of tidyverse is actually the balance between simplicity and modularity. And so in case you want to go out of the common routes, you can, the creativity is your limit really with all the animation packages that are out there and so on. And so we are not showing the only way to do it. That's a way that we think is a great balance between modularity and kind of simplicity of use.
Thanks, Stefano. And in the interest of time, I might let you have a look at the next little section on your own, which is how you can plot individual expression levels for each sample. So doing what we call strip chart and taking advantage of the facet wrapping ability of ggplot, so you can create multiple plots. So here it's creating a plot for each gene, and you can see what that looks like from the website, and also create interactive plots. Because I just want to get onto the composition before the break, so that you can have a look at that there. So more plotting ability. And with TidyBulk, just to mention, a nice feature it has is that it keeps track of which methods you're using. So we've been using methods such as edgeR, so it will keep track of the references for those methods, so to make it easier for you then to be able to cite them in any paper you're using or for your analysis or creating for your analysis. So that's what it looks like there. It's showing that we're using, for example, the edgeR algorithms, and they're the references handily generated for you by that get bibliography function. Okay, and the last part in the bulk RNA sequencing is just a little bit on this cell type composition analysis. So this is if you have tissue samples and if you want to see which different cell types are present, for example, immune cells. So for this example, we're going to use a different data set. So this one is from a large international breast cancer data set from what's called the TCGA project. And it's a sample of that data from the breast cancer, if I pull it in there. So it's got 22 patient samples from this breast cancer study, and it's got some transcripts, some genes from each of those patients. And what we're going to show is how you can detect which cell types are present in those breast cancer samples. So B cells, T cells, etc. So TidyBulk has this function called deconvolve cellularity that provides options for different methods. So CyberSort being probably the most commonly used algorithm. And what that will do is, if I run that here, is it will identify 22 different immune cell types that may be present in your samples. Okay, so what we should see when we do that is that we get columns output for amounts and proportions of different cells, T cells, and natural killer cells, monocytes, macrophages, etc. And what we can do then is, similar to examples we've shown previously, is we can shape this data using tidyverse style formatting. So pivot longer to get it ready to plot. So what this is doing is extracting the sample information, reshaping it so that we can then feed it into ggplot to plot these proportions of cells in each of the samples. So we've got our sample and we've got the cell type and we have the proportion of that particular cell type in that sample. And then we can quite nicely pipe that data, that format into ggplot and create a bar plot for the proportions. So here we are plotting our samples along the X, so each of the patients, and then what proportion they have for each of the cell types is colored. So here are the different cell types here. So quite a nice little plot. And we have the last challenge of this section, which is what is the most abundant cell type that we have in those breast cancer samples. So we'll let you have a little go at that and start that going. And so you can write some code that will figure.
Don't say the very easy way.
No, I won't. Okay, I'll say nothing.
If you can get it quantitatively, meaning looking at the actual data, amazing. Otherwise, you can use the lazy way. It's, you can imagine it's pretty easy. Even.
But yeah, I'll just give you a couple of minutes to have a go at that. Okay, 8 of you have already had a go. That's great. 9. And this then is the end of the first part, so the bulk RNA sequencing section. So hopefully this has given you an overview of how you can analyze bulk RNA sequencing data using these tidy packages. So tidy summarized experiment, if you've got your data in summarized experiment and you want to analyze it in a tidy way, you can use tidy bulk for performing bulk RNA sequencing analysis steps, and how these packages integrate nicely with the tidyverse. So using functions like filter and select and ggplot that already exist in the tidyverse. And also, excuse me, that you've seen how tidybulk also integrates different methods, so such as the jar, limma, DC2, all in the one package. And also if you're doing the cell type composition, it's got CyberSort and then a couple of other methods for that cell type composition analysis as well.
Yeah, just to point out that both tidy summarized experiment and tidy bulk are much more extensive that we have shown here. So many functions, both for the both packages that we don't have, of course, time to. So you can go on GitHub and on the README there is a full list of function for all these packages, and you can realize how extensive they are.
Okay, I might just give it to, what are we counting down to? 13:30, and then just end it there. So another 10 seconds. because then we'll give you the answer and you can have a break. Okay, the end. Okay, let's see. Oh, whoops, went too far. Ooh, it's a tight, close one this time. Okay, so the answer was M0 macrophages, if I'm not mistaken. And Frank, well done again. So that's 2 in a row. You're the out in front. So well done, everyone, though, for having a go. And I will give you, whoops. Okay, just to pop in, the solution is, get rid of that. This is one way you can do it, group by the different cell types and use summarize, tidyverse summarize to calculate the median proportion for the cell type, and then sort to see which sample, so macrophages M0 is the most abundant in that data set. Okay, great. So that brings us to the end of part 1. So now we'll have a break. So it's 20. So we just went 5 minutes over there. So we might come back at 5 past, 30 minutes. So 5 past the hour of whatever your hour is, wherever you are. And in part 2, then we'll have the single cell analysis part, and Stefano will take you through that. And you're welcome. People are putting thanks in the chat, and yeah, feel free to drop any questions in the chat that you might think of during the break. But otherwise, it's a nice chance to get up and stretch and yeah, get some water, anything. Okay, so Stefano, I might turn off my camera and what you call it, microphone, and then be back for 30 minutes. Okay.
Cool.
Okay,
Thanks,
see.
Maria. For many workshops, we provide the container as well. For this, I think we don't, because was not a requirement. But we have plenty of material online. We have done many workshops in these couple of years. Many of them have the container attached. And so all the recent workshops, the material is pretty similar. In some of them, we focus more on something or something else, but the baseline is constant. All right, let's get started. Welcome back, everybody. Thanks, Maria, for the heavy lifting at the beginning. I will show you now in this second section a bit of examples of how to perform analysis of single-cell RNA sequencing data using tidy principle that Maria has already explained to you. Just as very brief introduction, the difference between bulk and single-cell transcriptomics is that for bulk, we generally take a tissue, extract RNA, and sequence a biological replicate, for example. And we don't know which cell produce which molecule of mRNA that we are sequencing. That's why sometimes we use the convolution, because we want to infer back the composition of this tissue. With single cell, this information is quite more direct to us. And because the tissue gets digested, so we obtain single cell suspension, and we add a little barcode, a little tag to every molecule in each cell. And so then when we sequence our pool of cells, we know the transcripts that came from the same cell. And then we have actually a further dimension of information, which is cell ID. And we can have a much deeper understanding of on the heterogeneity of the tissue. And so as you can expect, having a total new dimensionality of the data, the possibilities are quite superior to bulk, and the analysis get quite more complex than bulk. Here is just an example of a very standard pipeline. We have raw sequence reads. Again, we can map them to the genome. We summarize, so we have for each gene a count. We can do data exploration as well before reducing the dimensionality of the data is very important to understand the property of our samples. We cluster cells in cells of similar transcriptional patterns. We classify those cells. We test hypotheses of, for example, if a biological condition has a different tissue composition than another. We can test again for differential expression, trajectory analysis, and so on. So quite a rich set of analysis. Okay, so let's go to the code again. Let's go back to the RStudio. You can open a menu here, as Maria showed you before. You can find the menu part 2, single cell sequencing, and it will bring you directly to the code, the line 832. Okay, so as before we start loading the needed library, there are, well, actually before, let me tell you a bit the summary of this second part. I will introduce a counterpart of tidy summarize experiment. As Maria showed, tidy summarize experiment is a package that allows to display and manipulate a summarize experiment object as if it were a data frame, and that allows open this object to the huge tidyverse and related packages. And in this part, I will show you two packages that do a similar job for single cell data, which is a tidy single cell experiment. And again, single cell experiment is an object from Bioconductor, a very popular container, and tidy Surat. And Surat is a workflow, but also an object, a data container, also very popular. And so we have created these interfaces for these two objects. There will be a different though. In the first part, we have shown you tidy bulk, which is a workflow as well. In this part, the workflow will be the same as the Bioconductor workflows and or the Surat workflows. Okay, so we don't provide a framework for it. The framework we will use will be the published one. So we will just show you how to and manipulate the data in a tidy manner. Okay. Let's load the library. There are a few libraries here. You can recognize some of them. Some of them, like Skater, are workflows, again, utilities from Bioconductor, some annotation packages, and so on. Okay, as before, we load our object. In this case, is a collection of peripheral blood mononuclear cells, PBMCs. This object is a single cell experiment, so from kind of the Bioconductor environment. This object would look like you have seen before for summarized experiments, meaning just a small amount of information is shown to you immediately, and you need to use ad hoc functions to extract and visualize the information. We obtain with a tidy single cell experiment that we have just loaded a very similar set of information as before. It looks pretty similar. We can see here that is a single cell experiment table abstraction. We have roughly 50,000 genes, and we have an assay that is count and log counts in this case. And you can see here some information relative to cells. By default, the the information that is displayed is relative to cells rather than and this is because cells for single cell experiment is very much a point of focus. So it's the element we analyze most. But as I will show you afterwards, we can join transcript information very easily for doing visualizations and summary statistics and whatnot. So you can see here that we have a cell ID. For each cell, we have a file. This file contains a database ID and index here. And we have some cell cycle information and the total number of counts for each cell and the total number of genes for each cell. As before, since this is still a single cell experiment, we can query with the functions we already know that work for single cell experiments, like assay names, and we get what we expect. So there is nothing different from single cell experiment. The difference is that now we can also use tidyverse on them. As before, I will show some example of how to query this object using tidyverse commands. In this case, we filter. Well, we get 0. Let me see. Well, let's see something that makes more sense. Let's say we can filter RNA. Here you go. We can filter with the criteria that we wish. And in this case, we have a filter based on the number of features. We get just 6. Then we can select. Yes, I had updated this, but again, we can select the columns that we wish. In this case, cells and count ID. Again, we can mutate these objects. So, for example, we can transform to lowercase the origin ident, and then we can select these. I apologize. This code is a bit of earlier version than I thought, but basically you can manipulate the object and then select this information. We're simply transforming lowercase a piece of information here. And this is also possible with summarized experiments. As Maria showed you before, we can bind object as if we would bind data frames. In this case, we bind a PBNC to itself, which is quite pointless. But sometimes we want to integrate data, so this they can become quite powerful. In this case, we have duplicated cells that have been deduplicated, and we have double number of cells. We have a file column, as I show you. This is an example, a more concrete example of how to clean up our data set. And as I told you, this file path includes information, which is the data set, the database, and the sample ID, which is the database plus a group. And so we can use some powerful tidyverse function, which is a strat, to extract that information using a regular expression. And so we extract from file a column called sample, and a column from file that is called dataset and groups, without creating temporary variable or whatnot. So we can imagine we can do a pretty powerful manipulation in a pretty simple way. And we are selecting just those columns. You can see here, that's the information we have extracted from file. Okay, so now let's go show you a bit how you can interact and visualize this object. let's go to the actual analysis. So I would like to point out that this workshop is not about, is not a reference of how to do the single cell transcriptomic analysis themselves. Is just some example on how analysis and visualization in a tidy manner. So one thing we might want to do is some quality control and filtering. In single cell, we might have a fluidomics machine that takes every single cells and adds tags to them, and these single cells are included in droplets. But actually, these droplets might be empty, so they might contain RNA that was floating in solution, or might contain cells that are dying or are fractured, so on and so forth. So we want to eliminate all those type of empty droplets and dying cells. And I will show you here an example of how to do that. Usually this filtering is done per sample independently. And so I will show you just for a sample here, we can filter our object with for these data sets. We can see here this data set has just 300 cells, so it's pretty small. This is very simply bioconductor code, nothing very tidy here. We just are giving the gene names and getting the location of our genes. Some of the location will be mitochondrial. Now, why this is important is because a symptom of a dying cell is a high percentage of the RNA content coming from the mitochondrion for the biological process the cell is going towards. So that's something we use to filter for alive cells versus dead cells. Again, this is quite common for bioconductor. We are just extracting some metrics based on how many mitochondrial genes are being transcribed. We obtain a data frame. You can see here the sum. Quite important is mitochondrial percent we will use to filter. And you can see here the row names are cells ID. And what we do is simply to transform it to a table and add information about what cells are high in mitochondrion. And we use the function isOutliers also from bioconductor that applies some criteria to decide the threshold to apply based on the percentage of the transcriptome from mitochondrial genes. Again, we have our data set with statistics. We convert it to a table, and then we add a column that is called high mitochondrion, and that's what we get, exactly what we would hope. And we have high mitochondrion here is either true or false. And we want to filter, we will want to filter the cells that are high mitochondrial false. Something a bit more tidy here. It's very easy to attach information to our single cell experiment object with the left join. So we have this mitochondrial information that has cell ID as column, and also our data set has cell ID as column. So we are joining by cell. And so you can see here that we have our column that we had before, but now we have also the columns of our mitochondrial statistics. And normally what we would do now is to, let me see, okay, we will do in few lines. I can show you here that we can easily filter this data for high mitochondria. Let's copy for safety. And here we have our supposedly alive and healthy cells from this data set. You can see here that we have dropped very few cells for this data set. So if you have any questions so far, please put them into the chat. Now, the next topic is a bit complex. So I want to make sure everybody is up to date with now to then focus on the next thing. Okay, so.
Actually, Stefano, sorry, I have a question.
Yeah.
Here we are analyzing the mitochondrial amount. Is it in a data set, or is it usually a sample? So here we've got 10 data sets and 20 samples. So there's 2 samples in each data set. Do you normally analyze them by data set or by sample?
In principle, you analyze them by sample because they might have been sequenced and processed in a different way. So a sample could be more healthier than another. In a way, you want to calculate statistics per, processing run. This is a dummy data set in a way. Mm-hmm. And so I just, because the samples coming from a data set are quite identical. And so I process them by data set in this case. But yeah, usually you process by sample because you assume it might be biologically independent from the others and might have different.
Cool, thanks.
Okay, so can I ask you to write to the chat how many of you in your analysis have heard about the concept of nesting and mapping from tidytranscriptomics? You can answer yes or no. Right now, no. All right. Okay, so these so these are quite, I would say, fairly advanced concept in tidyverse, and I think also one of the most powerful one for replacing all the iteration we normally do in R with for loops and creating variable and updating variable in that sense. This is because R is at its core a functional programming language. This is very much a functional programming approach. Every time we want to apply a procedure on many elements or subsets of our data sets. So the concept is that we group some element in our data set that we might want to apply a sort of function on, and then we apply iteratively the function. All this in a functional way, but I will show you now. So let's suppose we have a table here, very simple. We have a label, which is either A or B and some value. And so what nesting does is to produce another data set that includes data set themselves. And visually it's pretty clear what I mean. Now we are nesting the column value. So we have our column label that it can be A and B. And our data column includes a table itself that, as you can see here, is grouping these A and B values. Indeed, if we pull the column data, we will see that is a simple list of data frames with one column in this case. Now, it's quite powerful because now we can either update these columns or create new columns and do complex information processing without ever losing any information about the grouped data. I will show an example here. For who is familiar with group by and summarize, this is a much more powerful concept. In this case, we are creating a new column called average. And we are using the map functionality from PER to apply a function to this column. In this case, we are creating a new column average, taking the column data and calculating the mean of its value. And you can see here that this is a summary value of the content of this column. Okay, let's see a bit more complex example. We have calculated our average. We can filter based on our average, and then we can unpack the data because we never lost this data information. And so we have done a quite complex operation without creating any variable, and everything is it's very self-contained and quite robust in terms of the bugs that we might introduce in the code. In this case, we have calculated the average. We have filtered the row with the max average. In this case, this second row, label B, and we nest the data. Here we go. So I will show you, let me see. Yeah, the last example of this dummy dataset. In this case, it's a bit more complex. We are calculating the average again. We are applying a map, so not taking just the data, but taking both the data and the average column, and we are subtracting them. And in this case, we are updating the data column itself. Okay, we are not creating a new column. And we are, again, unpacking the data, and you can see here the outcome. Now, in this case, we have calculated the average, and we have subtracted 3 and 8 from this value column. Okay, so in this case, we have created new columns, updated the old one, and unpacked.
Stefano, we have a question in the chat. Is unnest like ungroup? And it is, isn't it? It unnests in the same way that ungroups.
Yeah, it's similar concept, but once you do group by and summarize, for example, you lose all the columns you were not grouping by. In this case, you never lose anything. It is a bit more like a database in a sense, but it's exactly the same concept here. It's indeed nest, applying something and a nest is similar to group by, do something and ungroup, yes. So why we might want to subgroup our data set, apply independent calculation on our subgroups, and maybe merge all together. Well, I just show you that we might want to group our data set by sample, applying some quality filtering by sample, and then put everything back. Normally, we have to split the data, so create a variable with a list of splits, applying a for loop, and then merging back. In this case, everything can be done through a pipe, no variables created, and less chance to introduce bugs in our code. And in single cell, it's very common that we might want to analyze the data independently from every cell type we might identify, and so on and so forth. So it's if you think about that, you in your single cell analysis, you will find many opportunities to improve your code in that way. Now, let's apply to our data. Again, let's group by datasets. And tidy single cell experiment is quite powerful because it abstracts the same concept of nesting and unnesting and mapping to single cell experiment objects. And And so we have seen here that we have created many data subsets according with the dataset variable. And this is really flexible. We could subset our dataset very easily on any combination of any annotation. We can subset on PCA, we can subset of number of cells, we can subset of on anything. In this case, we want to subset by dataset because of our quality filtering. And now there is a challenge. Okay, that's another quite difficult one, but you just have to copy and paste the code to answer this. So I show you how to nest and unnest. You have the code above, and I show you how to filter data for one sample. You have the code above. Now the challenge is, uses the next nest sample above, can you create a column that includes the mitochondrial information for your PBMC object? Again, I basically show you all the elements. Now put together the elements and nest your PBMC dataset and create a new column using mutate that will include the mitochondrial information that I show you above. This is not on mentee. So I give you a few minutes to play around with the code, and feel free to post the, I would say to post the code in the chat when you are ready. Let's see if it doesn't get too messy, but we should be able to understand your code from the chat, hopefully. And then I will show you how to do it. Again, if someone needs a bit of help, I will refresh. You know, around here, I show you how to obtain the information that for every cell give us statistics about mitochondrial transcription. This is a block of code, and then below I show you how to mutate after nesting. So let's do this actually. Instead of posting your code, post write in the chat when you are thinking you are done, and maybe I will ask to someone if you want to post your code to share.
And Stefano, they're trying to generate that table that you have down below, isn't it? Like the further below.
Well,
further below, isn't it? Did you show the aim is to try and see if you can generate this?
Yeah, that's, you're right. Yeah,
That's
so,
the goal.
Yeah, exactly. That's good. Yeah, to point out. This is the how the table will look like. So we have created a new column that will include the mitochondrial info. Exactly. Thanks, Mike.
And yeah, so Stefano showed all the steps for one data set. And then the idea is, can you use nesting to generate it for all 10 data sets?
Scaling and normalization is also something is usually done per sample. But a single cell, this concept of grouping, analyzing, and ungrouping is very, very recurrent. How is going, everybody? Who is trying? Let me know if someone is trying, so we know how long to. Failing miserably, that's good. That's a step in the process. Yeah, it's not the most simple thing, but once you grasp it, you understand how your life will become much easier. Okay, I will wait 30 seconds. That this was pretty challenging, but if someone wants me to wait, let me know. Otherwise, in less than 1 minute, I will share the solution. Okay. All right. How to do it? I will copy and paste, so I won't make any typo and delay the whole thing. Again, this could have been done with copy and paste of basically the code I showed you before. Just put together a couple of concepts. Again, we have nested our data set. We have a column called data set and a column called data. As before, from this nested data set, we apply mutate. We create a new column. And through the function map, to the functionality map, we apply a procedure to every element of this data. And so before I was showing as the inside the map, you have this tilde that is saying that your application of your function is starting here, and this .x that represent the content of this element. If you do map on one column, so just data is .x. If you map on two columns, so you kind of put together information from two different nested columns, it'll be .x and .y and so on and so forth. So in this case, we are getting the column data, so map column data, and we are applying our QC metrics on the content of our data. Same as before. Basically, this is a copy and paste of our code before, and that's it, basically. Our new column that we'll call mitochondrial info will contain this process information. So if we proceed, mitochondrial info will be a data set I showed you before for every sample, and I can show you what that here. If we do slice, so take the first row and pull the content of mitochondrial info, you will see what's inside actually is exactly the same data set as I showed you before. Okay, so take this as reference. You can, again, the code will be online, go back to it and really master the concept, and you will thank us hopefully. So we have our data sets where we have our original transcriptomic data and our mitochondrial data. And what we do now, we take this and left join, so join the information of these columns. So basically merge the two columns. again, because the two columns have this cell ID, the two columns have this cell identifier in common, it's very easy to left join them. And again, we run iteratively through each row using map. And we use map2 that takes data and mitochondrial, which is represented as .x and .y. And we simply left join the information. We forget about our mitochondrial information now because we have that join in our data column. And that's it. Basically, we have in one go, in a sense, without creating any temporary variable, we have grouped the data, calculated some new information, joined this information, and ungrouped the data. And now if we unnest the data, we will obtain our familiar data set, but now our independently calculated quality control for every sample will be in these, as you can see here, high mitochondrial and so on, columns that we have. Okay, now let's see some visualization. When you do quality control, is very common producing some plots that visualize for every sample the distribution of, for example, total read count, a mitochondrial percentage, and so on, to realize which cell might be dead. Usually, dying cells have a high mitochondrial percentage and low counts. As you can see here, again, these are called volume plots in a sense, but B-swarm plots better. And there are many packages that produce them in one line of code, and you might be able to kind of customize these plots. But for real customization, again, we show you how to do it with ggplots that have really no limits with all the ecosystem that is around it. We reshape the data because we want to obtain a faceted plot in one go. And so we are interested in the three variables. One is how many detected features, the total count of RNA output per cell, and the mitochondrial percent, the three variables that might inform us about dying cells. And we color them by high mitochondria, so the variable we filter cells on. Apply some styling, and here you go. We applied color, alpha, so transparency, and so on, and size. And the red are the cells we will filter. As you can see here, are high mitochondrial and generally low count. Okay, all this was an example of how to do a quality control, and now we just filter cell with high mitochondria, with low mitochondria. If anybody has any question, I will stop for 1 minute, take a breath, and maybe think about if there is anything you didn't grasp or you want to know more about. All right. Next up, there is another very common procedure when we analyze single cell transcriptomic data, which is scaling, similar to what we have observed before, and integrating. So again, there is some technical variability that we don't want to be confounded from in our analysis. One is every sample and every cell might have been sequenced with different depths, and so these overall different total count might not something we are interested on in terms of biology. and also some more complex technical variabilities based on cell processing or whatnot. And so this is called scaling and integrating. There are quite complex concepts in single cell because it's more, many of them are not kind of linear operators. And so you can read your literature here is very, very vast. Here we show you an example of it using bioconductor. Again, we want to do scaling based on data set. And so we nest again based on our data set, as I showed you before. We apply multi-batch norm function from bioconductor on the same data. So we are just updating our data column and get all our normalized data set and plug them in an integration function, MNN, that will find anchors in this data set and represent this data set in, so to speak, the same space, adjusting for all technical variability. And then, because these functions are not really designed to be tidy, some information has been dropped, so we just left join all the previous cell annotation that we had before. But as you can see again, without creating any temporary variable, we are just grouping our data. We are just subsetting our data set based on one variable, applying some function, put them together and join some information. So for a few complex processing in one go. And you can see here our PBMC objects. We have, it's pretty similar to before, but we have these corrected columns that are reduced dimensions. So similar to principal component, so to speak, but now are in a common space for all samples. So the technical variability has been compensated for. And now we can analyze all cells together. So try to cluster cells and analyze the biology. So now that we have integrated our data set, we can identify cell clusters. So this is an unsupervised clustering, and it's very useful to try to identify how many cell subtypes there might be in our samples. Again, this is nothing about tidy. This is a quite common code for bioconductor. In fact, we are using something that is just bioconductor code that is completely compatible with what we are doing, because what we are using in the backend is actually a single cell experiment object. So anything is applicable to single cell experiment is of course applicable to what we are doing now. And we have just create a new column with this build SNN graph and a cluster work trap. We are extracting the cluster membership, so the cluster ID, and introducing it as a factor. And here we can see that the new column is called label. Now, every cell now has a label attached to it that is the cluster number to which it belongs. With tidyverse, we can create summary statistics pretty easily. For example, we might count how many cells there are in each cluster. We have 11 cluster. We are showing the first 10 here. This is the number of cells that are in each cluster. Pretty straightforward. But as you can imagine, we might do much complex summary statistics out of those in a pretty straightforward way. And now that we have identified this cluster, it's almost time to visualize them in space. As before, of course, we cannot visualize a very highly dimensional space. We have many thousand features, so many thousand dimensions. We want to reduce the dimensionality to generally 2, so we can visualize them on the screen. In this case, we use something called UMAP, which is a non-linear way to transform the data. It does a similar job to we have seen before principal component analysis, but as this data is such multidimensional, to express the local similarity of the cells as well as the global differences or similarity of the cells in a 2D space, PCA is not the best function. And so there are other functions, for example, UMAP and TSNE, that are very popular to better represent the data. Again, this function is from Scatter, from Biconductor, nothing tidy here. What I want to show you, applying this function, it automatically updates the underlying object, and we simply see 2 added columns here, UMAP 1 and UMAP 2, because we have asked for 2 reduced dimensions. And now again, we can use this new column to visualize, to create summary statistics and whatnot. Now, this is a somehow similar challenge that Maria posed before with the principal component analysis, and is without creating any variable. Again, we are using piping here. Check if the variability of the first UMAP dimension when calculated 2 components, so we are just doing here, we are calculating 2 components, and we have this UMAP dimension here. So check if the variability of the first UMAP is equal more or less compared when you calculate 3 components. So before we saw that we can calculate 2 PC principal components or 3. Similar things apply here. What I want to know is the range of the UMAP 1 stays the same, decreases or increases if you calculate 3 components. Again, please go to mentee.com as you were doing previously. And Maria, maybe you can assist on this as you have the control panel then.
Yeah, I've set it to that question, so people can vote now on Mentee. So the options are equal, more, less, or don't know. And we've a 3 people voted so far.
Yeah, take your time, execute the code. It's really easy to analyze the variability of a variable, in this case, UMAP 1. You can calculate the standard deviation of that variable, and does the standard deviation of that variable changes in the 2 conditions, whether calculating 2 or 3.
So somebody's got in the chat, it should be less because you include more component, yes. Well, we'll find out shortly.
Again, yeah.
I was just going to say, Stefano, did you mention TSNE yet? Or
No, I didn't mention.
Just in case anybody knows of that one and why you're using UMAP instead of.
Yeah, Disney is another very popular, especially at the beginning, dimensionality reduction method for highly dimensional objects such as single cell. And why UMAP is becoming more popular is because Disney models very well the locality, so the neighborhood of each cell, but doesn't represent at all the long distances. So if you look the neighborhood of cells, you can distinguish one setup to the other, but if you have, you know, a nearby cluster and the faraway cluster, it doesn't tell you anything about whether those two clusters, you know, are very similar or very different. It's a bit hard to explain without images, but UMAP preserves better both the local neighborhood as well the long distances in the 2D space. Okay, so I give just a hint for the last people who may want to try. I show you how to calculate the variability of UMAP1. So we are calculating UMAP with for 1 or 2 UMAP dimensions, and I can use summarize, for example, standard deviation UMAP1. Here we go. That's our standard deviation. It's pretty easy to do the same for a different number of components. Basically, I give you the solution, but please try nonetheless. Or another way to do it is you can pull that variable that it will basically create a vector with that variable, and you can pipe to the standard deviation function. It's basically the same thing. Amazing, they get a different answer. Well, it's huh. Okay. So 8, 9, 9. So we have a mystery. Well, I will investigate.
I think you can probably end it now, will I?
Yeah.
We've had a good number about. Okay, let's see. Okay. And the answer is more. Is that it?
Well, no, the answer is less. Could be less. Maria, can you confirm?
UMAP1 has more. UMAP1 of 2 components has more variability than 3.
Yeah, exactly. So
yeah.
So
that's how. So well done, Frank and Claudia. Well, Frank just slightly pipped Claudia. Very close race for the top 2.
And Fadel as well. So differently from PCA, which each component is independent to each other, for UMAP, if you calculate 2 components or 3 components, those 2 operations are not invariant for the component 1 and 2. So if you calculate more components, the variability gets distributed across components. That would not be obvious if you think about PCA. So well done to the people. And the mystery why UMAP it was giving me, for who is looking at the screen, different values for the standard deviation is that is not deterministic. So as an element of stochasticity in there. Also very different from PCA, where is very much deterministic. And nonetheless, generally speaking, you have yeah, the answer that if you calculate 3 components, the first component will include less variability. So well done to everybody. Just to give the option, we show here, as you might have thought, how to calculate 3 components. You simply change the number of these arguments. And now I show you something that you might not have seen very commonly. Since this data is organized in a tidy way and interacts with many packages of tidyverse, this includes Plotly, which is a very powerful package for interactive visualizations. And so, although we usually visualize the reduced dimension in 2 dimensions, what about if we want to visualize a 3D scatter plot of the reduced dimension? That's pretty straightforward with Plotly and our system. You have 3 variables. You simply state UMAP 1, 2, and 3. Pick up some colors, in this case, according to the cluster ID. And you can see here that, okay, this data set is quite small, but you can imagine if the data set is becomes a bit more complex, this can be very powerful indeed. Now my screen is very raw, low resolution. Of course, normally it's much easier to do this, but you can, of course, pen and explore these plots at your will. Very interesting. I found some relation and heterogeneity for the cluster that was not very obvious in 2D. In 3D, some relationship become pretty more obvious. So I suggest you to explore this possibility. Is a very cool option that is pretty easy to do actually. Yeah, exactly.
You saw the question in the chat, yeah?
Yeah, these are the 11 clusters. Yes, you can with this you can deactivate clusters to if the space is too cluttered, and you can again interactively explore your heterogeneity in this way. Very cool indeed. Okay, we have a few last topics. Yeah, in time. Okay, so can we do it for samples? Yes, you can do it for samples. Let's see. I hope the column name is sample, if I remember correctly. Yeah, you can do it for any variable, any combination of variables. Again, with this type of tidy organization, you can see here that the samples are generally mixed together, which is something you would hope. You don't want to that's why you integrate samples, because your technical variability coming from different samples have been adjusted for. So in this case, what we would hope.
Someone I thought was saying they like it. Yeah, a lot of people like that interactive Plotly view of the clusters here, Stefano.
How to not like it? Impossible.
Okay, and just letting you know, I added a screenshot of the leaderboard into the Google Doc that we've been using, just because you didn't get to see that last one, and I'll add it after we've got one more question left towards the end.
All right, so let's proceed. We have clustered now a obviously, obvious question is what each cluster does represent, which cell type it does represent. And so there are many ways to manually annotate clusters. And of course, there are automatic ways to do that. A common approach that I use, for example, is to use both of them. And now there are even more methods to mapping your dataset on already annotated single cell datasets through Surat. So obviously the research environment in this space is very alive. But here we will show you just a simple example as we are more focused on the tidy approach. Now, a manual annotation is composed by two steps. One, to identify which marker genes characterize each cluster. So in one cluster are generally highly transcribed than the other. And then use a heat map to visualize all marker genes for all clusters. And then, as I will show you, you can take those marker genes, have an idea of the heterogeneity on the heat map, and use your own knowledge or search the literature to understand what those clusters are. This is quite good because it doesn't depend on any, it's quite unsupervised in a way. And so if you have kind of uncharacterized cell clusters, that's an opportunity to characterize them. So it's not an incredibly nice heat map, but you can see here that, for example, again, this is not the best way to visualize them. You will have a higher resolution and bigger screens, but you can see here that you see the distribution of the cladogram of cells colored by cluster. You can see here that cells of the same cluster generally cluster together, even in the hierarchical clustering, as you would hope. And you have some heterogeneity here, and at this in this case, you would identify gene by gene and try to identify what each cluster is. Again, there are many heat map methods out there. This is from Skater, but it's just an example. In this case, we are not using a tidy. We are using a biconductor function on our tidy abstraction. So there is no problem with that, of course, as I explained to you more than once before. Okay, what about automatic? That's also a possibility. There are algorithms that, similar to the convolution, they take a reference signature and they try to understand what's the most likely cell type for every cluster. One of them is single R. And in this case, we are using as a reference Blueprint, which is a pretty big collection of transcriptomic signatures. What we are doing here is pretty simple. Okay, we are downloading Blueprint. Then we are taking the reduced dimension, the integrated reduced dimension. No, sorry, I mixed up variable names. We are taking the counts, plugging them in single R with this reference. And we are giving, in this case, we are letting know the algorithm that we have identified clusters. So the identity will not be attributed to each cell, but to each cluster. Here, our cluster. This outputs an object that we convert to a data frame and then to a table for a bit nicer visualization. And in this case, I'm selecting the label, which is our cluster, and the first label, so-called by single R, which is the cell type. And so pretty simple here, data set of 2 columns. We have a label in the first column and the cell identifier, cell type identifier on the second one. Can I determine my tissue? What do you mean? If your tissue comes from lung or liver or something like that? I would think so. In this case, everything depends on the signatures, on the reference signatures. There are many reference signatures that are tissue specific. So in a way, you would hope that the result for all clusters will be the same, of course, which might not necessarily be the case because in this case, you are analyzing single cell types, hopefully, while a tissue is composed by many cell types. So it's a bit strange thing to do in this case, but for sure there are ways to do it. Anyway, so now we have this annotation. And then as before, we can left join, which is again a tidyverse way to do it. Left join one data set to the other, and the common variable is labeled. Pretty simple. There is no room for error here. We don't have to match anything. And just for visualization, I show you the cell and the force label here and everything else you can see here. Easy. Okay, so now there is, I think, the last challenge. So we might have winners here. Possibly.
Yeah, this is the final one. Yeah.
So Maria, prepare the prize at this stage.
Okay.
The acknowledgement from us is more than enough. Which cell type is the variable is called force label, as I show you, has the largest number of cells? So counting which cell type has the largest number of cells attributed to it. Pretty easy. I showed you before how to count occurrences in a data set. You can, it's a function with a pretty self-explanatory name. You can browse the code up to find it. You just apply to this column. Again, enter in mentee and choose your answer. We leave humans for that. How are we going, Maria, with the answers?
6 people voted so far, so maybe give them another minute or so.
Yeah, and then I will show, that's a good idea to post the screenshot to the Google Doc, then I will show you. This is the last one, the past one, but I will show you the
I'll stick
your
that
rank.
in. And I think Frank was cake, so I think Frank has gotten them all, like been top for all the questions
Amazing.
so far. So well done, Frank. Can you keep your leadership? Claudia might pip you.
We have a new collaborator. I will tell you which function to use because of course mentee takes tracks of the time, so who it very quickly will be an advantage. Count as I wrote to the screen is your function. Pretty straightforward. So you are counting how many occurrences there are in whatever variable you are interested in. Any more? We can.
I would say just 10 more seconds.
Yep.
Oh wait, actually maybe I'll give it to 12:30, so 15 more seconds. 13, 12, 11, 10.
That's stressful.
So I can see the countdown. 6, 5, guess. Okay, I'm gonna end it now. Okay. Oh.
All right, share it on the screen.
I will keep the suspense. Mentee's doing its calculations. Okay, here we go. Posting it into the Hey, can you see?
I can see.
Hey.
That's amazing. Dolphin, Claudia, Shaman, Dolphin Wood. Yeah, also, yeah, the first three, very solid. Yeah, also Basis. Yeah, very good. There are many participants. So the first that are displayed are very much very performing. Yeah, I think the ranks stay pretty same. Amazing. All right, thanks. The right answer, well, Paddle also get always the right answer. But is Bissell the right answer? Well, I might show you how to do it. Pretty easy. We have PBMC. The function of predictable explanatory is count, and we want to count what was the variable first labels. Very easy. And then that's it. We could. What is not B cells? Well, let's do it properly, and then we can arrange all these are tidyverse function arranged by dash, so descendant order n. So is skeletal muscle. No, B cell is not unfortunately this time the right answer. Skeletal muscle cells, and then NK cells, and so on. Good. Good. All right, amazing. Well done, everybody. Well, we are you can see select cell first label, everything arrange well. No, the question you see I'm answering to Fadel that posts the code. So the variable n count RNA is the total number of RNA output for each cell, while the question was relative to which cell type includes the most number of cells in a way. So every cells have been given a cell type. So in a way, we are counting the number of rows that correspond to each cell type. But very good attempt. I can see you nonetheless did pretty similarly the thing I was, we were looking for. So very good. Okay, so we are going toward the end. The last thing I wanted to show you is a pretty neat thing as it combines single cell and in a sense bulk analysis. And often both for data exploration and for hypothesis testing, for example, differential expression, we do what is called pseudo bulk analysis, meaning that we have 15 plus years of development of method for bulk RNA sequencing data. And so sometime we group our single cell data together to form as if it were a bulk RNA sequencing data, and apply some methods that we know how to perform, that we have done benchmark over and over, and then we are very familiar with. For example, you could, if you have a sample, you have many cell types, you could group by cell type and then sum. So basically unite all cells in a sort of purified tissue, if you like. That's an example, and then you can compare, cell type transcription from different samples, so on and so forth. Okay. So that's the concept. We sum the transcription of many cells to obtain a pseudo bulk, and then we apply operation to it. So I will show you just one example here. Here again, we have our original data set, and we created a function that is also present online in my gist in my GitHub that I will give later on, but it's also present in this package, in this repository under the R script. If you use it, please mention us, that would be nice. It's pretty simple function. We apply it, and it's pretty cool function because it does what I said. So it groups cells based on file. So in this case, we are discarding the cell types and so on. And it produces, from a single cell experiment, it produces a summarized experiment. And of course, Maria showed you everything about summarized experiment before. So suddenly we can apply, stay in the same tidy framework and apply all the knowledge we have acquired. In this case, you see we have a few different files, and we can identify for each of those files that are actually data sets. We can identify abundant transcript and scale abundance, like normalize, that's the jargon used normally. Again, you can see here the scale counts. And again, we might, we can do cool things. For example, since we have a couple of minutes, I can show you, we are scaling, and then we can, of course, reduce dimensions. And again, we might plot those reduced dimension into the, in two-dimensional plots, and see which of those samples are more similar to each other. In this, when you have single cell, sometimes it's very hard to understand it. If you do pseudo bulk, it's so much easier to plot those samples, representative of those samples in reduced dimension, and understand that my treatment samples that have been treated are more similar to each other than the negative controls and so on. But this is also used for differential expression analysis, but we don't have much time now. Some other material that we have produced show this. Anyway, this was just for showing that this exists, and this is the nice thing to plug single cell into bulk infrastructure seamlessly. Okay, the very last thing I will show you is the counterpart of tidy single cell experiment, which is tidy Seurat. Single cell experiment and Seurat are two data containers that are be different from each other. We created packages for both of them in a way to create an consistent interface. And so if you have a Seurat object and you load tidy Seurat, suddenly this Seurat object it will look again like a table. That's in this case a Seurat table abstraction. Again, you have your transcript and assays and so on. And as before, you can apply all the function we know. You can apply Surat specific function if you're familiar with Surat, and you can basically use the Surat workflow. Or you can apply tidyverse specific function as before. This applies, we can filter, select, join, bind, extract, unite, and so on and so forth. So it's very cool that you don't need, in a sense, to at least to manipulate these objects and visualize them. You don't necessarily need to learn twice the vocabulary specific from each package, but this is all tidyverse vocabulary that now is becoming really common knowledge. So you get, in a sense, many things for free here. Okay, so that's it for this single cell part. These are the key points. Feel free to read them, but it is basically what we have been described so far, what single cell, a tidy single cell experiment in the Surat are. Okay, Maria, I might give you the word to you to the concluding remarks. I hope you guys enjoyed. That was very good that many of you participated. Can we give the winner? We know who is the winner. Probably Frank is our winner, but you can see here the rank. Congrats to everybody, and very good.
Little clap. And yeah, thanks everyone for coming along. And I do need to point out that there is a feedback survey from ISMB. Stefano, if you wouldn't mind showing that link. So in the feedback section here, in the R&D, there's a link to the survey for the workshop. And it's also in the Google Doc link that we've been sharing, that we put in the chat, that has Frank's winning screenshot.
That's very important, because we always change, adapt the workshop based on the feedback. So all our
so yeah,
has impacted a lot our choice of material and so on. So let us know.
So please let us know anything you liked, anything you think could be improved. Be great to get that from you. And we still do have, now we penciled in this last, I think we've got even 30 minutes free for people to ask questions. So if you've got anything that you want to ask about bulk RNA sequencing, single cell RNA sequencing, tidyverse, the things we've been showing, if you want to go back and review anything, now is your chance. And you can feel free to unmute yourself and ask it if that's easier than trying to type.
So there are two questions. One, questions are coming. All right, let me see. How can we identify? All right, how can we identify the parameter? For example, how many principal components that we need to use, or how many K? All right, interesting question. Those are important parameters. Usually, bigger is the information content of a dataset. So huge dataset might need many more principal components to be described. For example, a UMAP and clustering, most of them are based on principal components. They don't work directly on the transcriptomic data. So there are functions to identify the optimal number. Now I don't remember the function, but if you Google it, you will find plenty of documentation. But basically, a very empirical way to do it is you can plot the histogram of variance explained by each component, and you will see that it will trace a curve. But basically, at the elbow, you have an idea of after a certain number of principal components, you don't add so much information to it. But there are actually statistical tests that do that quite more quantitatively. And how many K? Again, quite complex question. So the K for the graph, I assume you're talking about the neighbors. I don't want to say something wrong. But all those parameters, let's say how many neighbors you are considering for doing the graph. Again, if you consider more neighbors, you are capturing a bigger neighborhood, so it will become smoother. And the other way around as well, if you consider a small number. But there is a lot of literature, and it's not a straightforward answer. It depends on many things, including your data set. But very good question. Does tidy Surat have more feature than Surat? Well, no, because they are two different things. So Surat, in this case, is a analysis framework. Tidy Surat is just a data abstraction. Okay, so the two complements, they have different roles. Indeed, you usually use both, of course. So tidy Surat adds a link between Surat and tidyverse. So they are very two different things. Tidy Surat doesn't do calculation. It just helps you to manipulate the data using tidyverse.
And Stefano does have a paper on tidy Surat out now, which I think is linked in the material, but it's in is it genome biology?
Bioinformatics.
Bioinformatics, yeah.
We have also a paper on tidy bulk, which is on genome biology. This one. Feel free to have a look. So will sample code in the RStudio session be available? The sample code, I don't understand. Do you understand, Maria? What's the question about?
So the material is available online, and I wonder, do you mean the actual R? Do you mean the R Markdown document? Is that what you mean, Josh? Yes, so the R Markdown. So actually, Stefano, can you go back to the web page that has, for the workshop, that has the link to getting started and then local?
So is in the
workshop introduction. Oh,
is this here?
No. Yeah, there.
Well, there. Page.
Yeah, if you go here, just to show them how to get to it, and go to the section that's in workshop introduction, and then go to getting started, the second last from the bottom. Getting started.
Here you go. Yeah.
Getting started, and then go to local. If you want to install on your own computer, see instructions here. So under local.
Yeah.
So just to show them what happens if they click on that link, if you want to, that link.
Instruction, I see, yeah. Okay.
So you can follow that link, and then this shows you how to install it all on your computer. And then do you see the bit underneath the code block that says to run the code? You could then copy and paste the code from the workshop vignette, or that link, then the R Markdown file takes you to the R Markdown file in GitHub. Perfect. Okay, he gets me. So click on that R Markdown.
Yeah, there are many ways to.
Yeah,
many ways to
get the code. But yeah, that's got it all ready to go. You can import that R Markdown file and then work with that.
For the TBMC Surat, did you have to do something to convert to tidy Surat? No. So the beauty of both tidy single cell experiment and tidy Surat, that they don't do anything to the Surat or single cell experiment object. They just change the methods, how to, the R displays to you, and as well they provide all the adapters to tidyverse. So so you don't have to do anything, because nothing happens to your object. Your object is still identical.
So all they have to do is type library tidy Surat, and then it becomes the Surat object becomes tidy Surat.
Tidy-like. And there is you can read the documentation. There is an option if you want to use tidy Surat but still display the old way. You can set up an option for that. You set up options, the global variables in R, and you can choose that, the old style visualization, but you will be used to manipulate using tidyverse still. Thanks. And just out of curiosity, for the pseudo bulk analysis, can you do it's at different level of granularity. Yeah. You can, yeah, very interesting. I didn't, we talk about nest, again, very powerful since we are here. We are aggregating cells. As Anne was suggesting, you can nest group as you wish and aggregate based on the group. So now we are aggregating, well, let me show the simple way before. We are aggregating on file. You could aggregate on cell type, for example. Well, this is not here because we haven't predicted. But yes, you can definitely group at which level you want. Yeah. No limits, any combination of variables that you want. You could even nest your data set, aggregate within the groups, and join everything together. Everything is very modular. Pseudo cells. I mean, can you do it at different levels? Yeah, I think I answered to you, but if not, let me know. If you could post the material before the session. Yeah.
I think but I think the material was supposed to have been sent around by email from the ISMB organized. So normally we would send it a few days before the session. So apologies if you didn't receive it, if you only got it now. Yeah, we usually do try and get it to people prior to it. But feel free to contact us through GitHub, the workshop. Actually, you're able to show them how to post an issue if they want. So if you want to play around with it more afterwards, and if you have questions or issues, you could email us. I think our emails are on the document, or else you can post an issue on the workshop that Stefano is showing you now.
How to contribute?
Yeah, it's in the contributing section at the bottom. Yeah, under the feedback.
Here you go. Yeah.
So that's one way to get there.
It's all in the website. Yeah. So
there's a link there.
Everything there. And all of this is of course, in GitHub. So we use issues for any issue. And then where is it? So here you go. You create a new issue. Well, that's valid for every package. I mean, I might post my GitHub as well, because there are all the repositories we have talked about are in here, and actually much more. My research is not only just about frameworks. And you will find all repositories there, open issues as much as you like.
Okay, thanks, Claudia. Claudia has got another workshop after this one. Yeah, so feel free to go if you like, but if you have any more questions, also feel free to stick them in the chat or to unmute and ask.
Yeah, and I will give you the questionnaire at the end, please. That's super important. Yeah. You don't need to do it now, but thanks, everybody.
Yeah, thank you again for attending.